Hello, and thank you for taking the time to join the Arthritis National Research Foundation first in a 5-part webinar research spotlight series. I'm Emily Boyd-Storman, the CEO, and along with the board of directors, staff, and those participating in the panel, I'd like to welcome you to the spotlight on osteoarthritis. Moderating the discussions today are two esteemed members of the ANRF Scientific Advisory Board, Dr. Martin Lotz of the Scripps Research Institute, and Dr. Anne Marie Malfait of Rush Medical College. Please help me to welcome Dr. Lotz.
The first presentation will be given by Michael Joynek. Michael is actually a current ANRF grant recipient. Michael works at the University of Utah, and he is there in an environment which actually is really very strong in working on genetics of diverse diseases. And obviously his project here is about genetics of osteoarthritis. Michael?
Great, thanks, Dr. Lotz. Can everyone see my screen?
Yes.
Okay, great. So thanks for the invitation to speak at the Researcher Spotlight Series. It's really an honor to be a current ANRF scholar, and I'm really excited to tell everyone about my lab's approach to identify and functionally characterize the genes and pathways that, when disrupted, can cause osteoarthritis. And our goal is to use this large data set that we have to develop therapeutics that potentially can limit the progression of OA. And as Dr. Lotz and Malfait have already said, OA, as everyone knows, is a serious disease. It's very painful. It affects a lot of people. And we have no drugs that inhibit the progression of disease. And the question is, why is this? So we do know that OA is caused by biological changes to cells and tissue of the joint. Yet, one reason we don't have any effective drugs is that we lack really a complete understanding of the genes and pathways that are responsible for the initial biological changes. So if on the left here, we have a healthy knee joint that's composed of many different tissues, and it's really unknown as to really what those genes and pathways are that initiate a healthy joint to go down the path of disease. And so if we can identify these genes and pathways, we can then design drugs that may change the course of the disease. And so from this, the question is, how do we identify these genes and pathways? So we use the power of families to understand how genes contribute to the development of OA. So here at Utah, the University of Utah, we have this one-of-a-kind database in the US that allows us to identify these really large families that are affected with OA. And so what we have here, this is an example of a large family. Affected individuals are marked in black. And so we can find these families within our population. We can then recruit them to our study. And then we can do genomic analysis to identify the genes that's responsible for the osteoarthritis in these families. Then these genes become good candidates for therapeutic intervention. We've analyzed a lot of families to date. We've analyzed approximately 150 families with different forms of OA. This includes foot, shoulder, and hand OA, among other forms. And we've had really good success at identifying many interesting candidate genes, as seen in the table below. And so now these candidates become these are good candidates for future study. And while our human studies have been very informative about identifying these genes and pathways, they really don't tell us how these genes cause OA. And it's really important to understand how these genes cause the phenotype, because that allows us to design better therapeutic intervention. And so to test the function of these genes, we use different animal models. And given that we have over 150 candidate genes, the first thing we need to do is determine if the function of these candidate genes in each of the families is actually disrupted. So to do this, we use one of our model organisms, which is a zebrafish. to quickly test the biological activity of the disease gene. So we can, depending on what the gene is, we can tailor specific assays in the fish where we can do both gain of function and loss of function of the disease gene and see how that affects specific readouts. So this is really a nice assay that says the gene we identified in the family actually has disrupted function. And once we have this data, we can then move to a more appropriate model system, which is the mouse. And we use the mouse because the mouse has joints that look just like ours, and they also develop OA just like us. And what we can do in the mouse is we have really, really fascinating and interesting techniques that allow us to specifically make mice that have the human mutation that we identified in families. And this is just one example of a gene we studied that's involved in inflammation. So we've taken that mutation we've identified in the humans that have the OA phenotype. We've actually made that mutation in a mouse. And then this allows us to compare a normal mouse to a mouse that has the disease gene. So this is a schematic of a knee joint, and you can see this is a close-up of the cartilage. In a normal mouse, when we induce OA, they have a little bit of loss of cartilage, so it's a pretty mild OA phenotype. But then when we look at the mouse that carries the human disease gene, we now see that the extent of damage and severity is significantly increased. So this really tells us that the gene we identified in one of our families really is sufficient to drive, to cause increased OA susceptibility in an animal model, in a mouse. And then once we have this mouse, we can then do other studies to examine exactly how this gene is functioning and in what tissues this gene is functioning, which then again will allow us to really rationally design therapeutics that may have a significant benefit on preventing or delaying OA. And so once we have all our candidates and have made some animal models, our goal in the lab is to really to identify drugs that limit the progression of disease. So once we've identified genes like RIPK2, for example, we could then do high throughput screens to identify drugs or chemicals that can modulate this pathway and hopefully either prevent or significantly reduce the rate of progression of OA. And so just in summary, I've told you how we've taken a kind of a multidimensional approach, starting with studying human families that have osteoarthritis to identify candidate genes. We use functional assays in the fish to predict how these genes we identify in families, if they have altered function. Then we move to more appropriate mouse models. And then together, this allows us to identify really multiple genes in a single pathway that might contribute to disease. So common pathways. So these are common pathways for therapeutic intervention, which allows us to identify potential drugs. And then given that we've already generated these mouse models, we can then rapidly take these candidate drugs we have, test them in our mouse model before we move on to human studies. And just quickly, I'd like to thank my lab, a bunch of people who do really interesting work, funding, particularly from the ANRF, and I have a bunch of collaborators who are great to work with. So thanks for your attention, and I'll take any questions. And I know I went through things fairly quickly, so if you have any questions, please reach out to me. You can find my lab site by Googling me. Thank you.
That's terrific. Thanks so much, Michael, for giving us an overview of your really unique approach. So what was so appealing about Michael's application when we reviewed it was, in fact, that he, I think he's unique in that sense in the country, that he has access to this unique resource at the University of Utah to do these genetic studies. But not only that, then he has the entire pipeline in place where he can use a simple model, the zebrafish model, and then a more human relevant model in the mouse to actually determine, which is the critical step in his endeavor, the function of the candidate genes that he identified in maintaining joint integrity or protecting against osteoarthritis. And then going all the way and then actually finding drugs that would hit on this particular pathway. So thank you very much for that, Michael. So please raise your hand or just speak up and unmute yourself if you have any questions for Michael.
Thank you so much, Dr. Lotz and Dr. Jernick. We do have one question, which comes from Isabel, and she asks, how long does it take to develop the OA once the mice are injected with it?
Yeah, so that's a good question. So mice normally develop OA after a long time period, so greater than a year and a half to 2 years. So we do we induce OA in the mouse either through surgery or injury, and that speeds up the process. So we do that, and then we typically look anywhere from 4 to 16 weeks after we induce, we perform surgery on the animal. So it's relatively quick compared to the human phenotype, which allows us to actually be able to screen more genes and do a lot more experiments. We don't have to wait 2 years to the mice develop a phenotype. We can do it with relatively short time period.
Great. One other question from fellow ANRF scholar, Dr. Zhu. He says, nice presentation, Mick. One small question for you. For the mutations you identified, do you also test if the mutations function differently in different joint issues and may contribute to OA at different magnitudes?
Yeah, that's a really interesting question. So we do our functional analyses in fish to show that these are coding variants that we find. So we can test how that coding variant affects protein function. And then, yes, a lot of these, for example, the RIPK2 case, it's ubiquitously expressed in all tissues of the joint. So we are now generating, we have actually generated animal models where we can activate that disease allele in specific tissues of the joint. So we can look at cartilage, bone, immune cells, all the tissues where we have the appropriate drivers, and see where that gene, that activity of that disease allele is necessary. So yes, we are starting to do those experiments. And yeah, I think it's really important to understand the tissue specificity of these alleles, because I think it's going to help in designing therapeutics. So yeah, thanks for that question.
Fantastic. Thank you for that. Another question, how do you identify OA severity in the family tree, i.e. the people to analyze?
Yeah, so the Utah population database is really interesting. So we have, once we get approval through the appropriate institutional review boards, we can actually search medical records of everyone in Utah and identify specific forms of osteoarthritis that they have. We then recruit those individuals into our study, and once we recruit them, they give us permission to look at their medical records or their x-rays. And so it allows us to verify whether the diagnosis was correct, how severe their OA is, and it also helps us identify other family members that we might have missed through the population database. So it's actually quite a unique resource. People had the foresight to set up the system in the 70s, and as far as I know, this is the only database of its kind in the United States. There are others in the world, like the UK Biobank and the Decode in Iceland, but it's really a unique resource, and people in Utah are really open to research. So people really want to know why they have the disease, and so the participation rate is very good. And people are very open about sharing their health status with us.
Wonderful. And last question for you: does the genetic knockdown model you showed undergo surgery to induce OA?
Yes. So in the RIPK2 mouse that I showed there, we do either a surgical model, which is destabilization of the medial meniscus, or we do a non-invasive model. Given that this gene is involved in inflammation, we've tended to use the non-invasive model where we rupture the ACL ligament, and in both models we get the same phenotype. We are interested in aging, and those experiments take a little bit longer. So we're just completing those. So we've aged the mice that have the disease allele for 24 months to 2 years, and we're currently analyzing all that data to see if they are also more susceptible to age onset OA.
All right, thank you very much, Michael, and thank you very much for all your questions.
Thank you.
So it's my great pleasure to introduce our next speaker. It's Denis Evsenko. Denis is also is a former ANRF grant recipient. So I think it's more than 10 years ago. In the meantime, obviously, Denis has established himself as a very independent and highly productive investigator in the osteoarthritis field, but beyond, because he has a basic platform technology in stem cells. Denis is at the University of Southern California. He's the vice chair of orthopedic research and directs a stem cell research and translational program. Denis is also an entrepreneur, and he has kept good contact with ANRF throughout the years, and we are grateful for his giving us time today. Denis.
Unmute myself. Can you hear me?
Yes.
And slides are displayed well, right? Well, thank you, Martin, and thank you for inviting me to speak today. I'm a former alumnus of Arthritis National Research Foundation, and it helped me a lot in my career development, and I'm really happy to come back and continue those interactions. So we had great three introductory speeches about osteoarthritis. So I don't need to spend a lot of time on that. So what I want to do today is to tell you a little bit about the new drug that is actually entering clinical trials very soon. And this development was based on a completely new principles. And therefore, we have some hope that this drug will be efficacious. So one of the important things that I want to mention is that osteoarthritis is not really just a disease of joints. It's part of this very complicated aging phenotype. And you can see that if up to 30% of adults develop it, it means that there are some really fundamental issues that actually predisposed to this disease, and we now are pretty confident, and we can say that this is just one of the diseases of aging. In many cases, not all. In some of them, there may be exceptions, such as major trauma or really bad genetics, but a lot of people will have it, and it's a systemic disease. It's a systemic disease that affects multiple systems in our body. It can affect any joint, but the most common ones, as Anne-Marie mentioned, is the three joints that are probably most clinically relevant, are the knee joint, hip joint, and also hand joints. And you can see that the slide just illustrates how significantly, exponentially, this disease progress with aging. And actually affecting more females than males after the age of 65. I mentioned that it's a systemic disease, but what I mean by that, and this is a very important concept that actually lays in the center of our discovery. Aging is now considered as is an evolution of many systems, but one of the central things that we are facing when we age is a syndrome called chronic low-grade inflammation. It's very different from this inflammation that patients experience in autoimmune diseases, such as rheumatoid arthritis. This low-grade inflammation that progresses with age, and you can see the 7 pillars of aging here, such as problems with stem cells, metabolism, function of macromolecules, they all go and are all connected through this nexus called inflammation in the slide, because what happens with us when we age is that a lot of cells in our organs, they undergo irreversible damage. So they become sick. And it's very different from a typical acute injury when you, for example, injure your skin or, you even break your leg. It's an acute local situation, and our body knows how to deal with that. Here, this problem is developing everywhere in our body, and it mobilizes our immune cells to try to clear it and resolve it. But unfortunately, it's so significant that often our body cannot, and our immune system cannot really resolve this problem. So it's ongoing for many weeks, months, and even years. So putting your organism into this low grade stress that continues and slowly damages multiple tissues, including your joints. And that's the fundamental difference between acute inflammation, again, that is actually designed to protect us and has a lot of good features, such as activation of stem cells, activation of metabolism. So you can see it's a very short red wave or damaging wave in acute inflammation when you have negative things, but then it's replaced by this green wave that is shown here, a regenerative wave, where all this regenerative, protective, healing mechanisms kick in, and actually the outcome is positive. And the molecule that is actually controlling it, one of the central receptors that controls this shift from damage to regeneration, is called GP130 receptor. It's one of the central immune receptors that controls this balance. Without going into too detail of science, I just want to mention that in acute conditions, and especially in young organisms, this receptor is primarily activating modules that drive healing response. This receptor is complex, and it actually has more regenerative modules within this signaling structure, and also modules or pieces of this receptor that can activate pathways that can damage and kill cells. So in acute conditions, it's primarily activating regenerative modules, and in chronic conditions that last for years, such as chronic osteoarthritis, it primarily activates modules that actually drives inflammation, tissue damage, and fibrosis. So that's the concept of this modular regulation, where good and bad come together and are very context specific. And that's what distinguishes our technology that we developed from everything else in the space, because a lot of drugs that were previously developed to target GP130 or interleukin-6 family of cytokines that activate this receptor were acting as on-off switches. So they were actually switching off both good and bad signals downstream of this receptor. What we developed is a very unique modular drug mediators that can actually selectively switch off modules within this receptor. In other words, we can selectively turn off destructive modules, but we can let the positive pro-regenerative healing modules to signal. And that way we can actually harness this regenerative power of inflammation, because inflammation is extremely powerful regenerative response. And that's the outline of the technology in a nutshell. So you can see that in chronic conditions such as aging and, as Anne-Marie mentioned, obesity, this chronic inflammation is very pronounced, and your regenerative signals are not sufficient to actually overcome it. But in the presence of our drugs, we hope, this is not something that is definitive, but what we hope, and we have evidence for, that we can promote this regenerative signals and change this balance, shift it to more favorable clinical outcome. And this is not a fantasy. All these principles were already published in some major journals, and it's actually applicable not only to the field of joint regeneration. We have shown now that it's applicable to other organs, including the skin, for example, where we can actually promote activation of regenerative signals with the small molecule approach. And just to show you one piece of data from the most recent large animal study, because before you go into human trials, you really need to test your technology in large animals, and it was actually done independently by Dr. Makhatik in Canada in a blinded fashion, so he didn't really know where's placebo, where's the drug. We gave him these vials and he conducted the study, and we were actually delighted to see that the drug had really major, and the drug I'm talking about is, we call it CX11. It's an injectable drug that is injected in the knee. We had a major effect on structure, an anabolic effect on cartilage itself, but we also suppressed osteophyte formation, and osteophytes are those spikes of the bone that are actually causing a lot of pain. And our functional behavioral assays have shown that there's a significant improvement in pain score. And it was actually even evident at very low doses of the drug, which is really interesting. So with all that, we submitted it to the FDA in 2020 and received very favorable feedback because the drug has very safe profile and because it's extremely selective. And at the moment, we are finalizing IND and safety, IND enabling safety and toxicity studies, which are required for safety purposes to actually go in humans. And we plan IND filing in about 6 to 9 months from now. And that's where we plan to start the trial. And you can see the team of people who actually will be running it. So it will be directed by Dr. Nancy Lane, who is a pretty renowned expert in the field of osteoarthritis research, clinical osteoarthritis research. And the reason why we selected Dr. Lane is not only because she's excellent and had multiple trials conducted in the last few years, but also we don't want to have this conflict of interest. As inventors of this technology, we want an independent person from UC Davis to direct it and to really run the objective, thorough evaluation of these results. And this is the team of people, a couple of orthopedic surgeons, Dr. Lieberman and Dr. Petrigliano, who will be doing physically these injections at USC. And this is basically the outline. It's pretty standard. Martin, I have given you a very sophisticated, actually, explanation why those trials are so difficult, because outcomes are really not as robust as in many other fields. But we are trying our best. It will be phase 1 to 8 trial, so it will be safety and preliminary efficacy combined together, about 64 patients, dose escalation, and that's a different readouts and outcomes that we are proposing. So this is my last slide, and I just wanted to share with you those exciting findings and mention that Arthritis National Research Foundation is behind that. I was funded only a few years ago, and now we have a real clinical candidate delivered for our patients. We hope it will be successful. So thank you for your attention.
Yeah, thanks so much, Dennis. So obviously Dennis is the prime example of what we would hope for in an ANRF funded recipient. Actually, advancing his career and actually really advancing, in fact, the field of osteoarthritis, bringing out of his lab a drug to a clinical trial. I think that's kind of the ultimate intermediate success, and we certainly wish you a successful journey through the clinical trial endeavor. So are there any questions for Dennis?
A couple of questions, yes. First of all, is this a single dose or will it be multiple doses?
Well, for this first trial, because safety is the most important thing at the moment, so we plan to start with a single injection. So it will be a single injection for 24 weeks. We will just keep monitoring the patients. Later the treatment regimen may evolve, but at the moment it's a single intraarticular injection.
So we have a bunch of other questions, but I'm afraid we only have 12 minutes left. And I think we really don't want to shortchange Rachel Miller's presentation. And then I'll turn over to Anne-Marie to introduce Rachel to us. Thanks, Dennis.
Thank you.
Thank you, Dennis, and thank you, Martin. And sorry, I disappeared there for quite a while. I only managed to come back on now, but just in time to be thrilled to introduce my longtime colleague, Dr. Rachel Miller, who's an assistant professor here at Rush. and has been studying mechanisms of pain in osteoarthritis for the past 10 years, and was at some point also the recipient of an ANRF grant. Rachel.
Thank you, Anne-Marie, and I'm glad we got to hear some of your talk, although unfortunately not all of it. So hopefully I'll capture some other aspects regarding osteoarthritis pain for our audience here. So we heard some nice basic work from Mick, and Dennis told us how we can translate this technology all the way to a clinical trial. So I'm going to take us back to the basic end again and talk about how we're trying to identify potential new targets to take the drug translation along regarding new treatments to treat pain specifically. So I have nothing to disclose. So we heard the common complaint of osteoarthritis patients is pain, and as a result, it's a leading cause of disability worldwide. And so the formal definition of pain is that it, under normal circumstances, serves as a warning signal. So when you touch a hot stove, as an example, this tells your body to quickly pull your hand away before you cause more damage to your hand. But under chronic disease circumstances, nerves get continuous stimulation, and this can go on to cause what's known as chronic pain. And this is no longer helpful, but is something that we would like to come up with treatments for. So this is what our lab has been focusing on. And just to give you some examples of how we're using mouse models to study this process, we can incorporate different fluorescent markers in our different kinds of mice in order to track how the nerves change over the course of osteoarthritis. So we can actually see the nerves changing their location and growing and sprouting in different locations as the mice develop joint damage in their knee joint. And these nerves actually change functionally how they're responding to different stimuli over the course of osteoarthritis. So the nerve endings are located in your knee joint, but the nerve signal travels along the nervous system to the dorsal root ganglia where the cell bodies are located. And then the pain signal propagates to your spinal cord and gets transmitted to your brain. So we can actually watch the neurons in the dorsal root ganglia respond as different stimuli are applied to the knee joint. So in this case, we applied a pinch or a mechanical stimulus to the knee joint of the mouse. And so then we can see the neurons responding or flashing in real time as the mouse is responding to this painful signal. And so our long-term goal is to develop new therapies that can prevent or delay the development of persistent pain. And so one question we'd like to answer is which molecules produced as a part of the osteoarthritis process inside the knee joint or other joints are responsible for this sensitization of the nervous system and ultimately chronic pain. So I'll give you one recent example here. This is a molecule called CCL2. It's a chemokine, so it can promote inflammation. But our work has shown that it can also signal directly to nerves. And so in clinical studies, people have found that the knees of patients with osteoarthritis contain higher levels of this protein compared to normal knees. And the amounts of protein present correlate with symptoms. It's also been shown to be predictive of future pain. So people that are currently not suffering pain in their knee joints, if they have higher levels of this protein in their knee, they're more likely to go on to have pain in the future. So this seemed like a potentially fruitful pathway to study in more detail. So I'll show you how we're using these different techniques that I just introduced to you to study how CCL2 in the knee can cause pain and if blocking it is useful in stopping pain. And so this is a mouse knee that has osteoarthritis. And so as I was showing you, you can see new nerves growing in the bone underneath where the cartilage damage is occurring, as well as new nerves growing in the synovium or lining of the knee joint where inflammation occurs. So you can see here in a normal mouse, you do not see any fluorescent signal indicative of nerves in these areas, but in the mouse with osteoarthritis, now you can start to see a nerve sprouting in both areas. And by using different types of mice, we can actually see that the receptor that binds this protein, CCL2, is actually being expressed by these new nerves that are growing in the bone of the mouse. So next we wanted to see, are these new nerves responding to CCL2 inside the knee joint? So now we can take a mouse, inject CCL2 into the knee joint, and then using our technique where we can watch the neurons responding, we can see in real time if these neurons respond to the CCL2. So this video is just showing you an example of that. So you can see this is a neuron, this round cell here, and the increase in green color is indicative that that neuron is actually responding as we're injecting the protein into the mouse knee joint. So a certain number of neurons will respond in a healthy mouse, but the number of neurons responding goes up in the mouse with osteoarthritis. And so now we wanted to go on to see if we can block this protein from signaling. Will this actually stop the mouse from having so much pain as a result of that process? So the way that we do this, we can just press on the knee joint of the mouse and determine how much force is required for the mouse to feel pain. So a normal mouse, you can go all the way up to 450 grams before anything happens. But the mouse, when they develop osteoarthritis, now the threshold of force goes down. So now the mouse is responding when you apply much less force compared to the control mouse. So when we inject a small molecule inhibitor that can stop the CCL2 from signaling, this in 1 hour time after the injection actually allows the mouse to go all the way back up to the control levels. So we're inhibiting this painful signal in an acute fashion, but the effect wears off over time. We also saw a similar thing a little bit later in the disease stage. And so our ultimate goal is to continue to study this pathway and see if we can come up with ways of changing how we're delivering the drug in the knee joint to prolong the effect. So after a single injection into the knee joint, instead of just lasting for a couple of hours, perhaps we can make this last for weeks. And so we're studying other pathways as well, but this is just to give you a general idea of how we can actually study the nerves inside animal models of osteoarthritis to hopefully come up with new drug targets in the future. So we have a lot of people at Rush, including my lab and Anne-Marie's lab, working towards this goal. And again, I'd just like to echo the importance of the ANRF support in doing this type of research. So thank you, everyone.
Thank you, Rachel. So you can see how the ANRF grant here has actually enabled Rachel to develop this new technique of in vivo calcium imaging, which will be really useful, not just to address mechanisms of pain in osteoarthritis, but also to test potential drugs and interventions. I was wondering if anybody had any questions for Rachel.
Thank you. We do have one question that came in. How does hot plate assay thermal hyperjuliasis compare to this assessment of pain by joint compression?
Sure, that's a great question. So when you're studying pain in different animal models, you can study how they change their response to thermal stimuli, hot or cold, as well as mechanical stimuli. So I showed you the example of mechanical stimuli. When you test osteoarthritis mice and to see how they respond to thermal stimuli, it's less common to see changes in response to thermal stimuli. That's more common in more inflammatory types of models that would be associated with diseases like rheumatoid arthritis.
Wonderful, thank you.
Did anyone have any other questions?
So just to give us a sense, Rachel, about the path forward, So you are trying to pursue CCL2 as a target for osteoarthritis pain management, and how would you do that?
So the receptor antagonist that we've been using seems like a potential way forward, but we'd need a way to prolong the effect. So we're trying to work with other labs that have ways of making the molecule either stick around longer or have a delayed release, so that you can deliver a larger amount and then the drug would slowly be released in the knee joint over time. We're more interested in a local release as opposed to a systemic release because CCR2 blockers have been pursued in clinical trials in the past, but through systemic delivery, and they have not been so successful for other disease indications, but they haven't been tested for pain specifically. So we're interested to see if that can go forward.
Thank you.
I think it's 1 o'clock, so I don't know, Emily.
Thank you. Thank you so much to all of our presenters and to our moderators today. There's definitely not a day that goes by that I don't learn something new about the incredible research that our scholars are working on. The word impressive comes to mind, but most importantly, I think it's the impact the research has now and will continue to have that makes us so proud of the work that we do and so proud of the scholars that you all had a chance to hear from today. We hope you can join us for the next Research Spotlight series, webinar, which will take place in 2 weeks on October 6th. We'll spotlight lupus that day. Look for a link on our website and in your email. Thank you again to our wonderful presenters and moderators for sharing their research. And most of all, thank you to you for joining the webinar, participating, and for always supporting ANRF. We hope to see you soon.
Thanks, Emily. Thanks, everyone.
Thank you for having us.
Yeah.
Thank you, Emily.